Hi everyone, and welcome to our video on some exam questions related to cell organization. Our first exam question states, after a meal rich in carbohydrates, the concentration of glucose in the small intestine changes. The table below shows the concentration of glucose at different distances along the small intestine. So as you can see, we have the concentration of glucose on the right, and then we have the distance along the small intestine in centimeters on the left. Now it says at what distance along the small intestine is the glucose concentration the highest. So which of these numbers is the greatest? It's 500. And what distance is that in centimeters along the small intestine? It's 300. So it's highest at 300 centimeters, and that would get you 1 mark. The next question says, use the data in the table to plot a bar chart on the graph below, label the y-axis and choose a suitable scale. So, first of all, let's see our table. So we need to plot the glucose concentration. So on the y-axis is going to be concentration of glucose in the units. So we're just going to write down concentration of glucose, and it's measured in mol dm to the power of -3. Okay, so we've got our y-axis labeled. Now we need to choose a suitable scale, so it goes from 50 to 500. Okay, so let's go up in 50s. So 50, 100, 150, 200, 250, 300, 350, 400, 450, 500. Okay, so we've done our scale. Now we just need to plot it. So 100 centimeters, it was 50. So at 100 it was 50. So let's just draw that bar. Okay, at 300 it was 500. So at 300 it goes all the way up to 500. So it goes all the way up to here. So draw a straight bar down. Okay, and then for 500 it goes to 250. So it goes to 250 here, and then at 700 it's 0. Okay, so we've done our bars. I've just highlighted them over here, and that would get you 3 marks. Okay, because we've chosen a suitable axis, we've labeled the y-axis, and we've plotted our bars. Okay, 4 marks there. Next question. It says, look at the graph above, describe how the concentration of glucose changes as distance increases along the small intestine. Okay, so if we go from 50 to 500, which is our y-axis, you can see that it has increased, and then from 500 to 0 it has decreased. And we can even interpret that from the table. So when it was 100 centimeters to 300 centimeters, the volume of glucose increased. Then from 300 to 700 centimeters, the concentration of glucose decreased. Okay, so how does the concentration of glucose change? So it increases from 50 to 500. That's what we originally see, and then it decreases from 500 to 0. That's what we have interpreted from the graph which we have just drawn. 2 marks there. Next question. Explain why the concentration of glucose in the small intestine changes between 100 centimeters and 300 centimeters. So if we go back 100 centimeters to 300 centimeters, we can see that the concentration of glucose has increased. So it's saying why has it increased? Okay, so why has the concentration of glucose in the small intestine increased between 100 centimeters to 300 centimeters? So basically, the carbohydrates have been broken down. The carbohydrates have been broken down into smaller molecules. They are dissolved into sugars. And you need to say how they have been broken down. They have been broken down by the carbohydrate enzyme. So what causes the concentration of glucose to increase? Because the large insoluble molecules have been broken down into smaller molecules which can then be absorbed. Okay, so the carbohydrates have been broken down into smaller sugars which can be absorbed. And how have they been broken down? They have been broken down by the carbohydrates or amylase enzyme. Okay, so they have been broken down by the carbohydrates or amylase enzyme. Okay, and that would get you 2 marks. Next question, 3 marker. Explain why the concentration of glucose in the small intestine changes between 300 centimeters and 700 centimeters. So if we go back to our table, 300 to 700, it decreases. So why does the concentration of glucose decrease? That's what the question is saying. Okay, so the concentration of glucose decreases in the small intestine because the glucose which has been broken down has been absorbed into the blood. Okay, so the glucose which has been broken down has been absorbed into the blood. Okay, now that would be worth 2 marks. It's a 3 mark question, so you will need to explain the process by which it has left the small intestine. So it's the movement of sugars, so it's going to be active transport. So the glucose which has been broken down has been absorbed into the blood by active transport. Okay, and that would get you 3 marks. Okay, next question. Plants transport water and mineral ions from roots to the leaves. Plants move mineral ions from a low concentration in the soil to a high concentration in the root cells. What process do plants use to move these mineral ions into root cells? So the clue there is mineral ions. Into root cells, they're moving from a low concentration to a high concentration, which means energy is required. And how does this happen? It's active transport. For 1 mark. Describe how water moves from roots to the leaves. So again, it's a 2 mark question. There are 2 keywords you need. It's saying describe how water moves. You need to state how it moves, what does it use, and what's the process called. So it moves via the xylem through the transpiration stream. Okay, so it moves via the xylem through the transpiration stream, and that would get you 2 marks. Okay, next question. So it says plants lose water through the stomata in the leaves. The epidermis can be peeled from a leaf. The stomata can be seen using a light microscope. The table below shows the data a student collected from 5 areas on one leaf. So we're given the data there. It says describe how the student might have collected the data. So we need to write a method for 3 marks of how the student could have collected this data. So first of all, look at the method. The epidermis can be peeled from a leaf. The stomata can be seen using a light microscope. And the table below shows the data a student collected in 5 areas on one leaf. So let's imagine a leaf. He has used the microscope to look up 5 areas on the same leaf and written down his results. So how has the student done this? Well, he's used a slide because he's placed the epidermis on the slide. Okay, so he's placed the epidermis on the slide. That's what he has done. Using the microscope, he has counted the number of stomata in one area. So using the microscope, he has counted the number of stomata in one area, and then he has repeated the experiment with 4 other areas. As you can see, there are 5 areas on the leaf, so he's repeated with 4 other areas, and as you can see, he has calculated the mean average. So calculated the mean average. Okay, and I've given you 4 points there, but you only need 3 to get the 3 marks. Now it says, what is the median number of stomata on the upper surface of the leaf? So upper surface, we need to work out the median. Now the median is the middle number, so let's order them from smallest to biggest. So it'll go 0, 1, 1, so 0, 1, 1, 3, and 5. So we've just ordered them. Now we can cross 1 from each side. So 0, 5, 1, 3. We're left with the middle number of 1. So what is the median number? It's 1 for 1 mark. Okay, final 2 questions. Calculate the value of x in the table. Give your answer to 2 significant figures. So where is x? X is the mean for the lower surface. So all you would do is you would add all these numbers together and divide by how many numbers there are. So you would do 44 at 41 at 40 at 42 at 39, and you would divide it by how many there are. There are 5 leaf areas, so you would divide it by 5, and that will get you the mean average. Okay, so the mean average will be 41.2. So 41.2, it says to 2 significant figures, so that would get you an answer of 41 for 2 marks. Now the plant used in this investigation has very few stomata on the upper surface of the leaf. Explain why this is an advantage to the plant. So if there are fewer stomata, that means that less water will be lost because there will be less spaces for the water to leave. So if there are fewer stomata, it means that there will be less water loss. So less water loss. That would get you 1 mark. If there is less water loss, what does it do? Well, if there is more water loss, it will turn the cells flaccid. And if there's less water loss, it will keep the cell turgid. Okay, so you could say turgid or prevents cells from wilting, or you could have said flaccid. Okay, and that would get you 2 marks. And that is it for this video. Thanks for watching. I hope you liked it. And one last thing, please subscribe, hit the like button and the notification bell.